We're going to spend the next few sections exploring these electrical properties in an individual cell in the nervous system, working from small to big, simple to complex. Although there are many different kinds of neurons, the same basic electric principles underlie their function. We'll start by looking at the electrical principles that come into play when the cell is at rest. If we look at a typical neuron, you can see that it has a variety of parts. A cell body, or soma, that houses the cell's nucleus and a variety of cellular machinery. Extending out from the cell body are fine processes called axons and dendrites. The outer surface of all of these parts is made up of a bilayer of lipid molecules that partition the inside of the cell from the outside. This is called the cell membrane. We'll be spending a good deal of time talking about each of these various parts of the neuron, but for now, let's just concern ourselves with a small patch of membrane somewhere on the soma. There's an awful lot of important stuff going on in this humble little patch of membrane. Even when the cell is at rest, it's not electrically neutral. If you measure the voltage, the difference in electrical potential energy across this membrane between the inside and the outside of a living neuron, you'll see a small but important voltage called the membrane potential. You might be familiar with the idea of potential energy or stored energy. A battery, for instance, generates an electrical potential or voltage between its positive and negative terminals. When we hook up a wire across the terminals, this electrical potential causes electrons to flow through the wire from the negative terminal to the positive one. We can use this flow of electrons to do work, such as lighting this light bulb. If we measure the potential difference between the inside and outside of a neuron at rest, we measure a voltage of about -70 millivolts. Voltage is a relative quantity. We can only really meaningfully talk about the voltage between 2 points. When we say that the voltage at a particular point in a circuit is a certain number of millivolts, we must also define what point we're calling zero. By convention, neurophysiologists consider the outside of the cell to be this reference, which in electrical nomenclature is called the ground. Voltages inside the cell are measured with respect to this extracellular ground. To give you some perspective, a AA battery has an electrical potential of 1.5 volts. So this resting potential in the cell at -70 millivolts is about 20 times smaller in magnitude. So the neuron has a small but not insignificant voltage across the cell membrane. How did it get this way? Well, first, let's state what it's not. It's not a battery like the AA we showed you earlier. Batteries like that generate voltages through chemistry, electrochemical reactions that release electrons on one side and that use up electrons on the other side of the battery. The key to understanding how neurons generate a resting potential lies in the stuff on either side of that humble cell membrane. Both the inside and outside of the cell are basically made up of water with a host of stuff: proteins, ions, and sugars. There are a whole host of charged ions and molecules floating in and around the cell, including magnesium, bicarbonate, phosphate, sulfate, and many proteins. We'll be focusing for now on just a few key players: the charged ions sodium, potassium, calcium, and chloride, which are present in reasonably high concentrations and are important in generating a resting potential in the neuron.